Already we had seen about the structure of plasma membrane, but in this video we will see about the detailed study of the phospholipid bilayer, membrane proteins, and the mobility of membrane proteins, and glycocalyx. Glycocalyx is nothing but the outer coating of the plasma membrane that is covered by some carbohydrate molecule. That molecule is known as the glycocalyx. Let's see. Plasma membrane. Plasma membrane mainly consists of lipids and proteins. This is the phospholipid bilayer, and this is a cholesterol molecule. Then carbohydrate molecule. That carbohydrate molecule is present upon, or it is coated on the plasma membrane. That molecule is known as the glycocalyx. Then this is the binding site of the integral protein, and this is the receptor protein, and this is the protein filaments. These are the protein filaments. This is the mainly it is a cytoskeleton in nature. It gives the rigidity, and this is the glycoprotein, and this is the extracellular fluid, and this cytoplasm inside the plasma membrane. And the plasma membrane mainly it function as a barrier. It is a barrier of compartments between two aqueous solution. Both aqueous solution. That compartments is present inside the cell. That compartment is present. The two aqueous compartment that is present inside the cell and out of the cell. The protein is embedded or inserted within the phospholipid bilayer. This protein molecule which is inserted within the two phospholipid bilayer. Mainly the protein molecule, mainly the function of the protein molecule is a selective transport of molecules and cell recognition. The phospholipid bilayer. The phospholipid bilayer, if you want to study about the plasma membrane structure, first we extracted the mammalian red blood cells, because mammalian red blood cells they did not contain nucleus and internal structure also. So we could easily separate a plasma membrane, or we get a pure plasma membrane for our studies. In 1925, two Dutch scientists, namely E. Gotter and R. Grentel, extracted the membrane lipids from a known number of red blood cells, because from mammalian red blood cells this is not contain a nucleus and internal structure also, so we could easily separate plasma membrane. That plasma membrane, that surface area of the plasma membrane, they identify the surface area of the plasma membrane. It consists of a monolayer of the lipid membrane. That surface area of the lipid monolayer which turn out to be twice layer. When it turn out to be twice, because when it is occupied with the erythrocyte plasma membrane, when it is occupied by the erythrocyte plasma membrane, the surface area of the lipid monolayer turn out to be a twice lipid layer membrane. So we concluded that lipid, it is not a monolayer of a lipid molecule. It consists of a bilayer lipid. So it looked like a railroad track appearance. By using a transmission electron microscopy, we could easily identify it looks like a railroad track. And we already know that it is a hydrophilic head group and hydrophobic tail group. Hydrophilic head group is a polar. It is a polar and it is a non-polar region. Hydrophobic, it consists of a fatty acid chains. What are all the phospholipids present in the animal cells plasma membrane? We have to see in this slide. The plasma membrane of animal cells contain 4 major phospholipids. What are the phospholipids? Phosphatidylcholine, phosphatidylethanolamine, sphingomyelin, and phosphatidylserine. These are the four major phospholipids which are present inside the or which are present in the plasma membrane. Phosphatidylcholine and sphingomyelin. These are the two phospholipids which are present in the outside of the cell. Then phosphatidylserine and phosphatidylethanolamine. These are present in the inner leaflet of the cell. Then there were a minor membrane which is present inside the cell, that is phosphatidylinositol. The glycolipid, which is a minor membrane, it is present in the outside of the cell. And the cholesterol is a major membrane. This is present in the both out and inside of the cell. Phosphatidylinositol is a minor membrane, but it plays a very important role in the cell signaling. Glycolipids is a minor membrane. Totally 2% of glycolipids present as a phospholipid in the plasma membrane. The cholesterol is a major membrane, and it plays a very important role in the plasma membrane. Then phosphatidylinositol and phosphatidylserine both are having the negative charge in the cytoplasmic matrix. Next, phospholipid functions. Phospholipids, these are mainly do the function. It acts as a barrier between two aqueous compartments, because this is the outside of the cell and this part is the inside of the cell. The phospholipids, it act as a barrier between these two compartments or these two aqueous compartments. Interior of the phospholipid bilayer is occupied by hydrophobic fatty acid chains. These interior of the phospholipid bilayer, they doesn't allow the water soluble molecules, ions and biological molecules, because it is impermeable in nature. Then second bilayer of naturally occurring phospholipids, it is a viscous fluid and not a solid. So the fatty acid of most natural phospholipids have one or more double bonds. This fatty acid contains one or more double bond, so it twice the chains and it make the hydrocarbon in a long way. So the long hydrocarbon chains of the fatty acids therefore move freely in the interior of the membrane, so the membrane itself so soft and flexible. The inside of the fatty acid molecule, it is having one or more double bonds, so these bonds can twist the hydrocarbon chains. So the hydrocarbon chain it make easily in a long way, or long hydrocarbon chain is making by these fatty acid double bonds. So the interior of the membrane, it can easily move and it is so soft and flexible in nature. Next, cholesterol function. At low temperature, the cholesterol prevents membrane from freezing and maintains membrane fluidity. At the low temperature, the cholesterol is react with the phospholipid bilayer and which is interferes, and they prevent the membranes from freezing and maintains membrane fluidity. At the high temperature of cholesterol, it becomes less fluidity and reducing its permeability to small molecules. Cholesterol and the sphingolipids. Sphingolipids is the composition of sphingomyelin and glycolipids. It forms the lipid raft. These are associated with specific membrane, namely proteins. These are mainly associated with the membrane proteins. Functions of lipid rafts. They are mainly do the function in cell signaling and the uptake of extracellular molecules by endocytosis. Endocytosis is the cell which are engulfing the external material. Mainly the cholesterol, they did not present in the bacteria, but in animal cell it is a major component. In plant cell they lack the cholesterol, but they have the sterols. What are the cholesterol do the function in the animal cell, they can fulfill by sterols in plant cell. Membrane proteins. Membrane proteins. What are all the membrane proteins? There were two membrane proteins are there. Integral membrane protein and peripheral membrane protein or extrinsic membrane protein. Integral membrane protein or intrinsic membrane protein. Peripheral membrane protein or extrinsic membrane protein. This is the carbohydrate molecule or glycocalyx. It is another way called glycocalyx. Then most plasma membrane consists of 50% lipid and 50% protein by its weight. Totally 5 to 10% of carbohydrate proportion of glycolipids are glycoproteins. Glycolipids and glycoproteins both are carbohydrate molecule which have only 5 to 10%. One protein molecule, that a protein molecule is longer than the lipid molecule. The one protein molecule that is equal to every 50 to 100 molecules of lipid. In 1972, Jonathan Singer and Garth Nicholson proposed the fluid mosaic model of membrane structure. Integral membrane which is inserted into the bilipid layer. The peripheral proteins are bind to the membrane indirectly by protein-protein interaction. It is very useful for convey the message of convey the message between the protein. These are mainly used in the protein-protein interactions. Singer and Nicholson distinguished two classes of membrane-associated proteins. What are they? Peripheral membrane proteins and integral membrane proteins. Both the proteins can be released or it can dissociated only by treatments that can interrupt or disturb the phospholipid bilayer. Disrupt means it can disturb or interrupt the phospholipid bilayer. How it can interrupt the phospholipid bilayer? First one, peripheral membrane protein. Peripheral membrane proteins were operationally defined as proteins that dissociate from the membrane by using polar reagent such as solution of extreme pH or high salt concentration. For a dissociation of peripheral membrane protein from the phospholipid bilayer, they can use such a solution of extreme pH or high salt concentration, but they cannot interrupt the phospholipid bilayer or they cannot disturb the phospholipid bilayer. Once dissociated from the membrane, the peripheral membrane proteins are soluble in aqueous buffers. These proteins are not inserted into the hydrophobic interior of the lipid bilayer. Already we know that peripheral membrane protein that is not present in between the bilipid layer, phospholipid layer of the membrane. It bound the surface. It bound the surface with the protein-protein interaction only. It is present upon the bilipid layer. So once we can dissociate, once the protein membrane, the peripheral membrane protein is dissociated from the solution that can be soluble in aqueous buffers, these proteins are not inserted in the hydrophobic layer of the lipid bilayer. Then instead of these insertion, they are indirectly associated with the membranes. They can indirectly associated with the membranes through protein-protein interaction. So these interactions frequently involve ionic bonds which are disturbed by extreme pH or high salt concentration. Peripheral membrane protein, finally they can interact with the protein-protein interaction and it can involve ionic bonds. These ionic bonds only disturbed by extreme pH or high salt concentration. Then these peripheral membrane protein, after that it can finally we can conclude that it involved in the interaction of protein-protein, between protein-protein interaction. Then portion of these integral membrane proteins are inserted into the bilipid layer. So they can be dissociated only by reagents or detergents. These detergents namely octyl glucoside, that solubilization of integral membrane protein by octyl glucoside. These integral or these detergent having amphipathetic molecule. This amphipathetic molecule means both hydrophobic and hydrophilic region is present. So this hydrophobic portion of detergents displace the membrane lipids and bind to the hydrophobic portion of integral membrane proteins, because the other end of the detergent molecule is hydrophilic. So the detergent protein complexes are soluble in aqueous solution. So integral membrane proteins are inserted into the lipid bilayer, so they can be dissociated only by reagents that can interrupt the hydrophobic interactions. The membrane spanning portion of transmembrane proteins. Most of the transmembrane proteins are integral membrane proteins, are usually alpha helices of 20 to 25 hydrophobic amino acid that are inserted into the endoplasmic reticulum when during the synthesis of polypeptides chain. These proteins are then transported in membrane vesicle. Through the membrane vesicle, these 20 to 25 hydrophobic amino acids of alpha helices transported from the endoplasmic reticulum to the Golgi apparatus and from there to the plasma membrane. Peripheral membrane proteins can dissociated by polar reagent, and if it is dissociation is occurs soluble in aqueous buffers, it is involved in the protein-protein interactions and it leads to the ionic bond. These ionic bond only disturb the phospholipid bilayer. Then integral membrane protein that are inserted into the lipid bilayer, it can dissociated by the detergent molecule, namely octyl glucoside. This molecule is an amphipathic molecule in nature. It is having both hydrophobic and hydrophilic region. One side of the hydrophobic detergent molecule is dissociate the lipid molecule, and then it is bind with the hydrophobic region of the membrane protein, because the other end of the detergent molecule is hydrophilic in nature. So it can disturb the hydrophobic interaction, and it can also soluble in the aqueous buffers also. Most of the transmembrane protein is a integral membrane proteins. This is alpha helices 20 to 25 hydrophobic amino acid. This is inserted into the endoplasmic reticulum during the synthesis of polypeptide chain. After that, it transported through the vesicles into the Golgi apparatus and from the plasma membrane. Next, first we see the peripheral membrane proteins. What are the peripheral membrane proteins? Actin, ankyrin and band 4.1. The peripheral membrane protein, it is a component of cortical cytoskeleton. It gives the definite shape to the cell, and that is by spectrin. Actin, ankyrin and band 4.1 is a peripheral membrane protein. This ankyrin serves as the principal link between the plasma membrane and the cytoskeleton. How it is giving the principal link? By binding the both spectrin and the integral membrane protein band 3. Spectrin is a peripheral membrane protein and band 3 is the integral membrane protein. By binding both spectrin and band 3, the principal link will be formed between the plasma membrane and cytoskeleton, that is served by the ankyrin. The ankyrin done a great job in the linking between the plasma membrane and the cytoskeleton. Next, this linkage between the membrane and the cytoskeleton, that is provided by band 4.1. Band 4.1 is the peripheral membrane protein. This linkage, it is formed by the binding junction. What are the binding junctions? Spectrin, actin and glycophorine. This glycophorine is also a integral membrane protein. by the junction of spectrin, actin and glycophorine. These also binding to form the linkage between the plasma membrane, linkage between the membrane and the linkage between the membrane protein and the cytoskeleton, that is provided by band 4.1. Integral membrane protein. What are the integral membrane protein? Glycophorine and band 3 are the most common integral membrane protein in the plasma membrane. This is the glycophorine structure, and this is the band 3 structure. It is the outside of the cell and it is the inner side of the cell of the plasma membrane. It is having the cytoplasmic matrix or cytosol. The glycophorine molecule, it is having 131 amino acid with a molecular weight of about 30,000, because glyco means carbohydrate, phorine means protein. Half of the molecule is having a carbohydrate and half of the molecule consists of protein. Glyco means carbohydrate, phorine, that is a protein component. So half of the molecule is having carbohydrate and half of the molecule consists of protein. It is having a 30,000 molecular weight. And a glycophorine, it is a single membrane. During the spanning, it is having the alpha helix of 23 amino acid. Glycophorine spanning, it is a single membrane. It is having alpha helix of 23 amino acid. What is the first transmembrane protein? Glycophorine. Glycophorine is the first transmembrane protein. Band 3 is the it is very useful for anion transporter. It is responsible for the passage of bicarbonate ion and chloride ions across the red blood cell membrane. This band 3 polypeptide chain is having 929 amino acids, and it is having 14 membrane spanning of alpha helical region. This glycophorine is a single membrane spanning of alpha helical region site, and it is a 14. This band 3 is a 14 membrane spanning alpha helical region, and it is consist of 929 amino acids. Next, the first transmembrane protein, that is namely glycophorine. Glycophorine, how the glycophorine to be analyzed by X-ray crystallography by using the bacterium Rhodopseudomonas viridis. By using this method, X-ray crystallography, we have to designated L, M and H. L means light, M means medium, H means heavy. According to their apparent sizes, L, that is light subunits. These are they are red color, and medium subunits are yellow in color, and heavy subunits, that is green in color. That light subunits and medium subunits each are having 5 membrane spanning alpha helixes. Both are having, or each are having 5 membrane spanning. Easily count, we can count 1, 2, 3, 4 and 5. Yellow color subunit, that is medium subunit, and light subunit, that is red color in nature. That is we determine as a red color 1, 2, 3 and 4 and 5. 5 membrane spanning alpha helixes. Both are each having 5 membrane spanning alpha helixes. And heavy is represented as a green color. It is having only one transmembrane, or one membrane, or a single membrane spanning alpha helix. Then this is the white membrane subunit. It is a peripheral membrane, that is binding upon the phospholipid bilayer. It is a peripheral membrane protein. This heavy subunit has only a single transmembrane alpha helix with a bulk of polypeptide chain. See this It is having the bulk of polypeptide chain on the cytosolic side of the membrane. This is the peripheral membrane protein. This is the light subunit, and it is a medium subunit. It is the heavy subunit, and it's a peripheral membrane protein. And this heavy membrane protein or a heavy subunit consists a lot of polypeptide chain or bulk of polypeptide chain on the cytosolic side of the membrane. Mobility of membrane proteins. Membrane proteins and phospholipids are unable to move back and front between the inner and outer leaflets of the membrane at an appreciable rate. But proteins and lipids are present in fluid lipid layer. So it move only the movement of the proteins and lipid is a lateral movement. After that, in 1970, Larry Fry and Michael Edidin, they can prove an experiment. They can fuse the human protein cell and mouse protein cell to produce the hybrid human mouse cell. They can easily identify if it mixed within the cell or not. They can easily identify with the fluorescent dyes. They can use fluorescent dyes for identification of human cell and mouse cell. For a human cell, this is an human cell. They can easily identify by fluorescent dyes. Then mouse cell, there are a green color protein component is present. They can easily marked by a fluorescent dyes. After that, 37 degrees Celsius of incubation, the human and mouse cell, they form a hybrid cell, and the proteins are partially filled in that hybrid cell. For half of the cell it is filled by human protein, and half of the cell it is filled by mouse protein. After that incubation period of 40 minutes, both the human cell and mouse cell protein molecules are intermixed. So it concluded that the membrane proteins can be moved freely in the plasma membrane. Next one, the glycocalyx. Glycocalyx, it is an extracellular portion of plasma membrane proteins. They are generally glycosylated. The surface of the cell is covered by a carbohydrate coat that is known as glycocalyx, that is formed by the oligosaccharides of glycolipids and transmembrane glycoproteins. If you get interested in this topic, please subscribe our Biological Garden channel. Thank you.